So I'm introducing our not final, but final speaker here in this series. It's Professor Romas Kazlauskas, and he has quite a diverse career. He got his PhD in chemistry, I believe, from MIT. And then we joined as a postdoc George Whitesides at Harvard. And George Whitesides, if you're probably not aware, that is the pioneer, I would say, of biocatalysis. Then he moved into industry for some short time before taking on a faculty position at McGill University. And Romas likes to explore other places, and he has done some visiting professorships at different places, like in Sweden and Korea, I believe. But most importantly, I met Romas in Stuttgart, where I was doing my habilitation thing. And I remember the first week he arrived. What did he write on our big white wall where everybody had to post their goals? I want to learn how to make 1 million lipase variants. And actually, I had to count the numbers to know what the number was. You asked me, teach me how to do genetic engineering. Yes. So he went back to McGill, but then I came to the University of Minnesota and I made it my task to recruit Romas to Minnesota. And now we have Romas here and he is organizing this conference. And of course, he's giving a talk about mutations. Oh, I forgot, there's another thing about Romas. He has a rule after him. The Kazlauskas rule that actually defines how the enantioselectivity of lipases and esterases, hydrolases in general, works. And I don't know if you're talking about this, but he's talking about how distant mutations make enzymes faster.
Oh, sorry. I'm very sorry.
Great, thank you. Thank you very much, Claudia, for that very warm introduction. I want to, an earlier goal was to make enzymes more enantioselective, and right now we've been trying to make enzymes faster. And I'm going to talk about how to make enzymes faster. And the answer is going to be there are going to be distant mutations that are going to be important. And the story that I'm going to tell you is about converting a hydroxynitrile lyase to an esterase. That'll be but I want to tell you a few and just show you a couple of industrial examples to connect this to industry. Here are 3 molecules that have, over the last decade or so, become commercialized, and because engineers were able to make those enzymes faster. So there are a few examples. So there's sitagliptin. This is the same example that Michelle Chang mentioned in her talk. And I like this as an example because it was a student that graduated from my group that was a first author on this engineering paper. So this is a drug molecule. And this is a DNA molecule. This might be something that you use. Twist Biosciences, this is a company that we use for DNA synthesis, uses enzymatic DNA synthesis, which is much greener than the traditional chemical method to synthesize DNA. So if you get synthetic genes or even DNA primers that are synthesized, they are likely made with an it's a terminal deoxy transferase. That's the key enzyme in this DNA synthesis. And finally, maybe you're wearing some perfume that is made from this. It's a molecule that used to be extracted from sperm whale, but now it's made synthetically with biotechnology. And I show a picture of the transaminase that's used here in sitagliptin. And I show you when they did this engineering, they used directed evolution, and they used about 15 or 16 cycles of directed evolution to find those substitutions. There's 27 substitutions in that enzyme. They had to expand the substrate range of this enzyme. And you can see where they are. They're all over the place in the enzyme. They're not just focused in the active site. And you know, my training as a chemist really wants to focus in the active site because that's where the action happens. So if you want to improve an enzyme, certainly that's where you should be looking, in the active site. But this is not true, right? This is the answer is not just in the active site. You're making changes all over. What are those mutations doing? And that's kind of what we're trying to show in this lecture. So first, a cartoon of what I'm going to talk about. So one of the things that a distant mutation might do is there might be some loop that needs to close and open in this thing. I don't have an example of that, and I won't talk about this example, but I will talk about these other 3 examples. So one of the things that we'll find is moving catalytic residues into the right positions. And you say, well, why are we making mutations out here? See, why don't you make the mutation right here? And it's because when you make a mutation, the smallest change that you can make is a methyl group or a methylene group. That's about, I don't know, 3 angstroms across or so. And you're trying to fine tune this. You don't want to move it by 3 angstroms. You want to move it by 0.3 angstroms or 0.1 angstroms. So making mutations too close it makes the changes are too big. You can't fine tune. So you make a change far away, and then you get propagation into the active site and make a small change. Same thing for binding shape. You don't want to make gigantic changes, small things. Again, make a mutation far away, and it will propagate it in. And finally, the other thing that we found are tunnels. You actually can create tunnels to allow substrates to go in and out or products to come out. And the example that I'll show you is going to have all 3 of those examples in there. So here's our enzyme that we're going to talk about. Let's start over here with the esterase, because this was going to be our goal. This is what we want to make. So an esterase, these are alpha-beta hydrolases that we're working on, and they have serine, histidine, aspartate, catalytic triad. I don't show the aspartate so it doesn't get too crowded in this picture. And they also have an oxyanion hole. These are nitrogens, main chain nitrogens that make hydrogen bonds to the carbonyl group, and they'll stabilize the intermediate oxyanion when the reaction happens. So let's just go through this. So it starts with the histidine, pulling off a proton from the serine. You have an O- here. The O- is a good nucleophile. It attacks the carbonyl. And the negative charge moves to the oxygen, and that oxyanion hole stabilizes it. So that's the first step of the reaction. And what else? So the example enzyme that we'll be working with is called SABP2, and it has a kcat under the conditions that we use of about 135 per minute. And we're going to use kcat to compare enzymes to say which one is better than another. And we're going to, over here, our starting enzyme is going to be, within the alpha-beta hydrolase family, most of them are esterases, but there are a few pockets of enzymes that do something else. And this is one of those pockets. So there's a pocket of hydroxynitrile lyases. And they are pretty similar. So they have a serine, histidine, and an aspartate, which I don't show. Same active site. There's a catalytic, there's an oxyanion hole here, but I didn't show it because there's a threonine here that blocks, there's a glycine over there. This one blocks the oxyanion hole, so it's not really accessible. And it catalyzes a completely different reaction. although it starts the same way. The histidine pulls off this proton, makes an O- here. I said this is a good nucleophile, but there's nothing to attack. So instead it grabs this proton, it acts as a base, pulls off this proton, and you create a double bond and eliminate the cyanide. And these enzymes are plant defense enzymes. The release of the cyanide deters the insect and keeps the plant alive for a little bit longer. And so there's a hydroxynitrile lyase from rubber tree that has this in the active site. And we are going to use, we're not going to work, for this talk, I'm going to tell you about a different enzyme. We're going to use something calling HNL1, which is an ancestral enzyme of the hydroxynitrile lyase from rubber tree. And we're using the ancestral enzyme because it's a little bit closer, about 50 mutations closer to an esterase. And we think our challenge will be a little easier to convert this into a into the esterase. So it's about 60% sequence identity. And here's the KCAT for that. It's about 100 times less, or maybe a little less, maybe. So, but it's definitely a lot less. So the first thing that we do, I think we had to do the organic chemistry experiment, and we said, let's do everything that touches. We're going to see that here's the substrate here, and we're going to make every single amino acid that touches or even has a chance to touch the substrate. We're going to make it the same, exactly the same as it is in SABP2. And that won't work. I'll show you the story here. So the black ones here, black blobs here surrounding this are all the mutations that we made. There's 15 mutations that we made. This is HNL15 all close. There will be another HNL15 in a little while. And it actually made it maybe a touch worse. So we change everything. The active sites are identical, and we get nothing, right? We're not getting any improvement in the esterase activity of this hydroxynitrile lyase. So we have to do more, right? We have to make mutations outside the active site. How are we going to choose them? We think they're not conserved, because if you just look at all esterases and all hydroxynitrile lyases and you look for conserved residues, you don't find things that are far away from here. And the reason for that, we think, are that there are different solutions. We'll see that what you have to do is push something or make a hole or do, so there's a lot of different residues or a lot of different ways that you can do that, so that the amino acids aren't necessarily conserved. So we still used sequence comparison, but a slightly more limited sequence comparison. So we stuck with a smaller family. We just looked at about 10 enzymes where there were some hydroxynitrile lyases and some esterases. And as well, we had reconstructed some ancestral esterases and one hydroxynitrile lyase. We have those 10 enzymes. We're only going to compare those. And the other thing we're going to do is we're going to rank the comparison based on esterase activity. So for example, this is a great esterase, 580. It's going to get a high vote in whatever amino acids it has in that one. And this one has got no esterase activity. It's going to get a 0 vote in whatever amino acids it has there. And there are other ones. So you can see the rates that we've measured for the esterase activity on every single one of those. And from this analysis, what we identified was 38 residues that were associated with esterase activity. And we made all of those mutations, actually with gene synthesis from Twist Biosciences. We made that variant, and it was a good esterase. But that's a lot. That's a lot to analyze. So we cut back. We first cut back to 16, and then we ended up cutting back to 15 residues. So we have 15 residues that make HNL1, this enzyme here, into a good esterase. Do I have a number here? Yes, I have a number. It's 100. Right? The KCAT is 100, so it's pretty close to 135. So we can say it's about as good as the esterase. And this chart says we need all of those mutations. So this is the catalytic activity of the wild type, which is the HNL115. And this is what happens if we take one mutation out. Just nothing. Right? Any one of those mutations you take out, maybe these two are a little bit more expendable. You only kill, go down in half or go down to 17%. But all of these others you're down to less than 10% of the activity. So every one of those mutations is doing something. And that's what I'll have to tell you. What do those mutations do? So let's first take an overview of the structure. So we have crystal structures of this HNL115. We have a crystal structure of HNL that we started with. So the first thing that we did was we compared them. I got a new enzyme, new activity. What does it look? Is it different? And so this one shows the differences. So this looks like B factors, but they're not B factors. This is telling you what moved, right? What moved in this structure. So this big red spot is the biggest movement. This is the largest movement, and these thin, if it's a warm color, it means a lot of movement. A warm color with a fat tube, it's a lot of movement, and these other thin tubes means nothing moved. The main chain is still in the same place. So this is a main chain comparison. And these black spheres here are the 15 mutations. So you can see that they're not at the edges, way far away. There are some on the surface, right? And some in the active site, as well as some outside the active site. They're really scattered all around. And I also have some answers here, what these things are doing. So I have these mutations are actually fixing an oxyanion hole. This biggest mutation is creating a tunnel into the active site. And these mutations over here, this movement associated here, is actually making more space in the binding site. So these movements are actually quite small, the adjustments to the oxyanion hole. So let's look, the next three slides we're gonna look at those three places. Here we go. So here's the oxyanion hole, and so the oxyanion hole is two nitrogens. So it's this nitrogen and it's this nitrogen. And the blue one is the SABP2. So that's our model. That's where we want to be. So you can easily see that the nitrogen here is in the wrong position, right? This is the white one is HNL1. This is HNL1. And when we did our mutations, we moved it a little bit closer to where it is in SABP2, but not actually that much. So I'm not sure how important this movement is. And then this one, well, the nitrogen seems to be in the right position, but there is a problem. It's the side chain. The side chain of HNL1, this cysteine, is in the way. It's blocking. This is where the oxygen has to bind. You can't put a sulfur over here, and you're blocking the space here. So you have to move this side chain out of the way. And what do you have to mutate? It looks like anything bigger, right? Here's a leucine here, and here's a phenylalanine in one of them. And if you make it a big group here, it's too crowded over here, and it moves out of the way, and now this is accessible. So one of the fixes is the fact that this has moved out of the way. I have a little animation here to show you this small movement. It's exaggerated a little bit, but it shows you that there's what this is showing is here's the nitrogen that's going to move, and there's green, 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 green. These are mutations, and it'll show you that everything moves. here. You can see that all of those things are moving together. It's not just one thing that just gets pushed out of the way. All of these mutations are changing their shape in order to move this nitrogen just a little bit. So that's one of the things that we fixed. So we pre-organize, right? We get the catalytic machinery in the right position, get this out of the way, and move this just a little bit closer. The next fix is the binding site. The binding site isn't big enough. So let's go look at this. So the natural substrate for SABP2 is methyl salicylate. And this shows salicylic acid, the product bound in there. You can see it a little bit there. And you can see that here's where the alcohol would bind. So there is space for an alcohol in this region. So the substrate that we're using is this with a large alcohol group. So there may be space here to put this alcohol. But if we look at HNL1, there's no space there. There's no space to put this group here. This is the natural substrate for a hydroxynitrile, and it would fit very similar orientation as it is right over here. And there's no space to put an alcohol. And here's our mutated enzyme with the 15 mutations. All right, we've created some more space there. So now the substrate can go in and it can fit this large alcohol there. All right, one more fix that we have, and that is tunnels. If we zoom out of these structures, what you see in SABP2, here's the active site here. You see lots of different tunnels in the top of this enzyme. There's especially a yellow one here, and there's a number of different tunnels that are coming out. But if you look at HNL1, you don't see a lot of tunnels. You just see one kind of tunnel in this area, and that's about all you have. When we made our mutations, right, we've created more tunnels. We've got a red one that you didn't have before, and especially this yellow one that you didn't have before. A tunnel, why would that help? Why do you need a tunnel for this reaction and not the old reaction? Well, for a hydroxynitrile, the substrate goes in, it gets cleaved, and it comes back out. Same way it came in, right? In and out through the same door. Esterases are a little bit different, right? The substrate goes in, all right? It has to go in with a paranitrophenyl group in first. And then what happens is that the serine can attack, and it releases the alcohol. So you have the alcohol that's released, and now this acetate group is attached to the serine. So this has to leave, but it can't go back out where it came in because, well, you've attached an acetate group here. You've closed the door is closed, it's locked. You have to go out a different way so this gets out of the way and to be able to put water here and to continue on the reaction. So creating an additional tunnel, we think, hypothesized, allows this paranitrophenyl group in order to escape. All right, before I have one last experiment to tell you about, and that is that we can apply this information that we've learned in order to make the natural enzyme better. So the natural enzyme here, SABP2, right? This is its natural substrate, and we are putting in this paranitrophenyl acetate substrate. And we thought, you know, accidentally, it might have enough room there in order to make in order to fit this large group, but maybe it doesn't. Maybe it doesn't have enough there. And one of the mutations that we made, it was at position 104. We put in a threonine at this position in our ancestral enzyme HNL1-15, our good one. And SABP2 has an alanine at this position. So a lot of esterases do have a threonine here. we thought, let's try this threonine. And what happens is that we got an increase by about 6.5. And we think what we're doing is making bigger space here for the leaving group, just like we did in the ancestral enzyme. So the same things that we learned from engineering H&Ls can help us understand what's wrong with a natural enzyme and make it also work better. So here's my summary slide. We started with an enzyme that was pretty bad, and with those 15 mutations that we put in, got to about 100 per minute. And we fixed the three things. We fixed the oxyanion hole, we made more space for an alcohol, and we created the alcohol exit tunnel, we think. And then we applied these ideas to fix a different enzyme, the SABP2, to make more space. And for that one, we ended up getting 900 per minute as the kcat. So we're really able to use that to make natural enzymes better. So the last slide is the acknowledgement. So my group is a group of biochemists. So we measured how fast we created those mutants and measured how fast they go and looked at those structures. We also collaborated with a computational chemist. That little animation that I showed you came from that group. And what they're doing is measuring how correlated motions can propagate from one part of the enzyme to another in order to move things. And finally, there's crystallographers here at the University of Minnesota, led by Bob Evans and Kei Shi, and a bunch of undergraduates that solved all the crystal structures. So there's two structures that I showed you here, but we solved structures for other ones that we have similar experiments that we have done. And finally, thank you very much for your attention, and I'd be glad to answer questions. Yes. Michael.
So all those 15 mutations made a lot of cool things, and you explained how sort of each one contributed to some sort of way to benefit the enzyme. But that tunnel, how lucky was that those couple of mutations created that tunnel? Like, was that just like divine intervention giving you a tunnel out of nowhere? I mean, how did that happen?
How'd that happen? I can show you how it happened. Or I can show you the two mutations that made that tunnel. Oh, I deleted them. They were two mutations, and we initially didn't think they're at the end of the tunnel, at the surface of the protein. And if you look in, you can't see them. And the mutations are actually to make the side chains bigger. Now that's what do you mean bigger? You're trying to make a hole, right? So we made them bigger, and what it did is it just moved the next loop over. And what you saw there in this structure, this is the loop that got pushed, right? These are the two mutations, and we made them bigger here, and they pushed this loop over, and that's what made the hole. That's where the tunnel happened.
Wow.
So for this type of analysis, like for these little changes, so you've been using crystal structures of proteins. I'm curious with like new advances with protein prediction tools like AlphaFold, how accurate you can predict these type of structure changes. Have you tried that? And if you're not at that stage, do you envision that that's going to be the future? Like, can we actually do this type of analysis without actually solving crystal structures?
So we had these crystal structures for a long time before we figured out what happened. We found the tunnel pretty quickly. But those, like the movements of the oxyanion hole, well, that's not the first thing we noticed, right? And the figuring out that the binding site changed, You know, it took a while to find that, right? So even when you have the crystal structure, you need to know what to look for. So, but AlphaFold, the goal of AlphaFold is protein fold. I want to get the fold correct. And that's often not what matters here. It's the fold, right? Those two folds are exactly the same, right? As far as AlphaFold is concerned, right? Even though the C alpha positions are slightly different and maybe the side chains are all different. So that's not what it's optimized for. So I wouldn't use AlphaFold for this kind of work. Although, I mean, I think when we solved that structure, we put in an AlphaFold structure as a starting point for sure. I mean, because it is a great program, but maybe not as detailed as these need to be. Bob has a question. Bob is the crystallographer that did all these structures. Did I make a crystallography mistake?
No, not at all. I just wanted to continue responding to the question here. AlphaFold was absolutely essential in our molecular replacement. So in order to phase the data set, we used AlphaFold. And it was good enough to give us these structures, but it was certainly not good enough to predict the structure. So we needed more. Okay.
Okay. All right.
Yeah, thank you for a nice talk. So your tunnel theory is right. Probably releasing a product might be the rate limiting step for of your enzyme, right?
So you're right. Leaving that's right. Losing the product could be the rate limiting step.
It can be confirmed by kinetics or something.
So we do know we have done experiments where you do what do you call stop flow experiments, where there was a collaboration with a German group that did that. And certainly the first step is fast, and the second step is rate limiting. But you kind of would have expected that, because this is a para-nitrophenyl group. It's a good leaving group. You really expect that first thing to fall off really quickly, and then the second step, the cleavage of the acetyl enzyme intermediate, to be rate limiting. So it's not a surprise that the second step would be rate limiting. So yeah.
Thank you.
Okay.